Well, we were all disappointed, I think, by these results. We were hoping for better results. The primary endpoints of death and MI and TLR were actually a little bit worse in the Absorb trial. Also, even the vasomotion just really surprised me. The vasomotion data was, if anything, a little bit better with Xience. Part of that reason is that the Xience stent performed pretty amazingly well. There's just such a lack of events. They actually both did well. It's just that the Xience stent did better as you look at it. So it's worrisome. This is an early stage device. I wouldn't give up on biodegradable stents, but I think we have to be very careful about it, and we also have to just be really careful about how we implant them. This idea of making sure we pre-dilate, we size, we post-dilate, we do imaging, and really get superb results and pick our patients carefully. This stent is still, I think, going to be useful, but I think the later iterations will be probably more helpful.
This stent is now on the U.S. market. It's available for routine use. You were making some comments a few minutes ago about how these data might somewhat alter your approach to whom you would use it in, how often you would use it. So if you could talk about that.
Well, the main issue, it seems to me, and I'm just digesting this, is thrombosis. So we're going to want to use patients who, we're going to use the stent in patients who are at the lowest thrombosis risk. That's going to be the younger patient who doesn't have renal failure, who doesn't have a calcified vessel, and has a larger diameter vessel. And also the other concept is to try and really get these stents well implanted without distal dissections and in large diameter vessels to minimize that thrombosis risk. And also use it in patients who have the most to gain from it, which are really the younger patients who have many years ahead of them. And in those patients with metal stents, you tend to collect a lot of stents throughout your life, and through their lives, I should say. And those are the patients that you really would prefer the stent to degrade and go away over time.
The stent, I guess, is degrading and disappearing, but one of the hypotheses was that there'd be a gain in vasomotion as an indicator of vascular healing, and that wasn't seen in these 3-year results, was it?
No, it wasn't, and it really shows you that you really have to test your hypotheses, and that theoretical ideas really need to be tested, because what Dr. Soroy's thinks is happening is that in between the struts, the stents, in between the struts of the metal stents, the vessel is able to expand, and that's what he's seeing. It's a very minor difference, but it was actually better. There was more vasomotion in the Xience stent, which really just shocked us, and it's sort of fascinating from a theoretical, intellectual point of view.
And one last thing, so from what I was hearing this morning, even as the scaffold starts to completely disappear as you go out to 3 years, there is still some residue that is thrombogenic that's left. They were talking about proteoglycans.
So the proteoglycans apparently, they're not endothelialized is probably the problem. And so they can be thrombogenic, and that may be one of the reasons why we have a thrombosis problem. And the other issue from the study from China that we saw today is that some of these events are happening on dual antiplatelet therapy, so it's not entirely clear that that's the answer. So there are concerns, but again, I wouldn't give up hope for biodegradable stents.